Hello, in this video we are going to cover chapter 11, which goes over meiosis and sexual reproduction, at least on the cellular phase. So one thing that you could do prior to watching this video is watch the Amoeba Sisters video on sexual and asexual reproduction to see what the differences are, because in this video we're just going to talk about sexual reproduction. So this is just a nice overview picture of what meiosis is, what occurs in a meiosis, and how the cells look at the end. Now I do want to point out that we end up with 4 unique daughter cells, and they are haploid. So I know the steps are going to look like mitosis, but meiosis is far different, and we're going to see that here. There are some nice worksheets that go along with meiosis. This one, Cells Alive, goes along with their website page. You can click and read about the different phases of meiosis. And then this one is just kind of straightforward. Name the phase, put the phases, or put the name with the phase here, and then identify some processes down bottom. So worksheets to keep in mind as you go through this chapter, chapter 11. All right, so let's get into chapter 11.1, the process of meiosis. We're going to describe the behavior of chromosomes during meiosis and the differences between the first and second divisions. We'll talk about the cellular events, and we'll explain the difference between mitosis and meiosis. And we'll talk about how genetic variation can come about during meiosis, which is very important. There is a nice Quizlet that goes over the vocabulary for meiosis. So I said back in the chapter 10 video as well that there's a lot of vocabulary in this chapter and in chapter 10, so it might help you to review that on the Quizlet here. But let's get into it. So sexual reproduction is not like asexual reproduction. Sexual reproduction requires 2 individuals to release their gametes, and then those gametes will combine to form a new, unique individual. So asexual reproduction, it only takes one individual, and that individual would make a complete copy of itself and that would be the offspring. So its offspring would be a clone. Whereas sexual reproduction, we're going to make a unique individual. At least one. So we're mixing chromosomes from 2 individuals that are different from each other in the first place, and then by mixing their chromosomes, we're going to make it an even more different individual at the end. So this process differs from mitosis because in mitosis, we're completely copying the cell, we're copying the DNA, like word for word, base for base, I guess you could say. We're copying the cell in mitosis. But in meiosis here, which is what we are concerned with this chapter, in meiosis, we are not copying point for point the cell. We end up, here's our beginning cell, and here are our 4 end cells that don't even look like the beginning cell. All right, so why is sexual reproduction so important? So mitosis is great. It creates an identical cell, which is good. We make a new cell from mitosis, but meiosis and fertilization is where it's at. So you get genetic variability from meiosis. Genetic variability is good because there's always evolution and adaptation going on in the environment, and you need to produce offspring that can beat or withstand or evolve or adapt to those different environments in different situations. So creating offspring that are more likely to survive because they have more variability, that's better than creating offspring that are exactly like you and maybe not as adapted to the environment. So offspring are not identical to one another. They're not identical to you either, visibly or not. So what do I mean by that? Well, you could look different phenotypically or physically, or you could not look different but still be different. So on like the genetic scale. So producing offspring by sexual reproduction creates a population that's less susceptible to disease, less susceptible to environmental influence. They're more able to adapt and survive. All right, so the process of meiosis. Most plants and animals have two sets of chromosomes, one set from each parent. So you have two sets of chromosomes for every pair, correct? Diploid organism is what you are. So number one, you have a pair. Number two, you have a pair. Number three, you have a pair, so on and so forth, all the way down. So each chromosome that you have in that pair, one came from your mom and one came from your dad. So 50-50 split. Now, it could be this first one came from mom, this second one's from dad, this first one's from dad, this second one's from mom. Mom, dad, you could say dad, mom, mom, dad, so on and so forth all the way through. But half of them came from mom, half of them came from dad. So mom gave you one copy of chromosome 1, dad gave you one copy of chromosome 1, okay? So that's humans, right? There are other organisms that have different numbers of chromosomes. You could have a triploid organism, which has 3 copies of each chromosome. You could have an octoploid organism, like a strawberry. They have 8 copies of each chromosome, which is insane. But 4 come from mom, 4 come from the dad in that case. So diploid, again, you're diploid. That means you have 2 sets of each chromosome. And then your gametes end up being haploid. And haploid is how we get to the diploid part. And that might not make sense to you right now, but it will in the future. All right. So in humans, meiosis usually produces what daughter cells? And I know this is probably preemptive, but you're going to produce, letter C, 4 haploid cells. 4 haploid cells. So remember, you have diploid cells in your body, but your gametes are haploid. So sperm or egg cells, they're going to be haploid, and you end up with 4 of them at the end of meiosis. All right, another poll question. If a muscle cell of a typical organism has 32 chromosomes, how many chromosomes will be in a gamete of that organism? So if their somatic cell, let's say they're a diploid organism, their somatic cell has 32, so what's half of 32? 16, right? So their gamete will have half of those. So they have 1 copy of each chromosome instead of 2 copies of each chromosome. All right, so the process of meiosis, reduction of chromosomes is necessary to avoid doubling in every generation. Okay, so I'm going to go back to our picture here of the chromosomes. So you get half of these from mom, half of these from dad, right? So your gamete is going to have 1 copy of each thing. So 1 plus 1 equals 2. Okay, so that's normal. If we didn't have haploid gametes, let's say we just had a cell like this, and it met up with another cell that looked like this, well, then we're going to end up with 4 chromosomes on each number here. And that's not good, okay? So we have to have a reduction. We have to cut our chromosomes in half so that we get 1 plus 1 equals 2 for each chromosome. So gametes or spores are produced from what's called a germ cell. The germ cell is what starts out meiosis. So we'll see what that does here in a minute. Excuse me. And gametes are going to be, again, haploid. So meiosis reduces the number of chromosomes by half to make haploid cells. So body cells, mitosis, this is how mitosis usually occurs, right? We start with a cell that looks like this, and then we end up with two cells that look exactly like that beginning cell. Okay, so that's what we already know about mitosis. Meiosis, on the other hand, we're going to start out with a cell that looks like this, but then we're going to end up with 4 cells that have half that number of chromosomes in it. Okay, so it would have 1 pink and 1 green at the end. And that could end up being an egg, or it could end up being a sperm cell. Okay, so normally I would do this activity with you all if we were in person. You're welcome to try it on your own. You're just going to make a nice little poster about a phase of meiosis. And all you do is title it with that phase. You'll draw, color, and label it, and then write just really shortly, briefly, what happens in that phase. And then we would normally present it. Okay, so where does meiosis begin? So this might look familiar to you. We already talked about the cell cycle. So meiosis is going to take the place of mitosis here. So the cell is normally going to have an interphase, and then bam, we're going to go into meiosis instead. So a germ cell is what starts out meiosis. Okay, so a germ cell is going to divide into into 4 daughter cells. Okay, so these are the phases. This is just the overview. We're going to go over these step by step here. Meiosis is split up into two stages. So we have the first division, which ends up with 2 cells that are haploid. And then we have the second division, which ends up with 4 haploid cells. So yes, you heard me correct. So after the first stage of meiosis, we already have halved our chromosome number. So we end up with haploid cells at the end of meiosis 1. So meiosis 1 is preceded by normal G1S G2. And then we start with prophase 1, metaphase 1. So the 1 is denoted by the phase of meiosis that we are in. Okay, so we're still dealing with chromosomes, we're still dealing with sister chromatids, but we're just going to line them up a little bit differently in this stage of meiosis. So this stage looks very different. Meiosis 1 looks very different from mitosis, okay? So prophase 1, we're going to start it out. We have our sister or our homologous chromosomes. So if this is chromosome 1, here's our 2 copies of chromosome 1, they're going to attach to each other. So normally this would not occur in mitosis, but this is very unique to meiosis. Okay, so they hook up in a synapse, and then they're going to exchange some portions of their genetic material, and that's called crossing over. So this is one way we can get genetic variation through meiosis. Again, this doesn't occur in mitosis. This is only for meiosis. They're going to exchange genetic variations in their gene loci. Okay, so it's only going to be on the identical parts of the homologous pair. So crossing over in prophase 1 is the first instance where genetic variation can increase. So we'll look at the other instances here in a minute. Prometaphase, so again, it's just like regular prophase. The spindles are going to connect with the centromeres. And then in metaphase 1, this is our second location where we can get genetic variation. So you might look at the image here on the top and the bottom and might say that those are the same. They are not the same. They actually end up with different cells, different daughter cells. So this is what I'm trying to show you with the genetic variation. The way that the chromosomes line up in the center at metaphase, if we think back to mitosis, normally they would make a single file line all the way across. In metaphase 1, they are with their homologous pair in a double file line all the way across the middle. So the way that that pair lines up, it could, let's look at this bottom one. It could be blue-red, or it could be red-blue. And then those chromosomes, so this blue and red, are going to get pulled to this side. This red and blue will get pulled to that side. And then that's very different from up here where it's blue and blue go this way and red go this way. Again, this is just for 2 chromosomes. Remember, you have 23 pairs, so there are a lot of instances where the flip-flopping can happen, and you can end up with some very genetically different situations here at the end. All right, so anaphase 1, the actual whole chromosome gets separated into a separate cell. So instead of pulling sister chromatids apart, the homologous pair gets pulled apart. So if this is chromosome 1 and chromosome 1, now they get separated. And then telophase 1, the cell will try to reform. It might not actually fully reform because it has to go into meiosis 2 here in a second. All right, so again, cytokinesis, how we separate the cell. In animal cells, we create a cleavage furrow with microfilaments, microtubules, and then in plant cells, we create a cell plate to separate the cells. Okay, so into meiosis 2. So we go directly from telophase 1 and cytokinesis to prophase 2. There is no interphase between the 2 stages of meiosis. Okay, so there is no interface between meiosis 1 and meiosis 2 because we don't want to duplicate our chromosomes. We want to have them, right? We want to get fewer. That's the whole point of meiosis. We're trying to get fewer chromosomes in our daughter cells. So there's no interface. So we go directly from telophase 1 into prophase 2. So prophase 2, again, no interphase, no interkinesis, never S phase. Chromosomes are going to condense if they started to unravel. They probably stayed condensed, and the spindles are going to start to reform. So remember, at the end of meiosis 1, we already had 2 daughter cells, and they are already haploid. Right, because this is chromosome 1 here, this is chromosome 1 over here. So both of our pairs, chromosome 2 as well, they got separated, and so now we have half the number of chromosomes. So at the beginning, we had 4 in our cell, but now we only have 2, right? Okay, so prophase 2, like that. Now we're going to separate the sister chromatids in meiosis 2. So spindles will reform, and then in anaphase 2, we're going to separate the sister chromatids now. And this is going to look a lot like mitosis. So meiosis 2 looks a lot like mitosis. And then we create telophase 2, cytokinesis, which ends up with 4 cells, right? Because now we're creating that cleavage furrow on those 2 cells, and then that ends up with 4. And so now with our crossing over that occurred, you can see the little red part there on that guy, the random alignment during metaphase 1, and then there is some random alignment at metaphase 2 as well. That's going to create a lot of different variants at the end. So, if you want to click through an interactive to learn more about meiosis, you can do that at Cells Alive. They have a really nice one, really simple. And so I want to point out, before we move on, I want to point out how the chromosome number gets halved. And so I've been saying this all along. We're going to start out with 23 pairs, right? Our germ cell, our body cell, it starts out with 23 pairs, which is 46 total. So after interphase, we're going to have still our 46, but remember, we go from that to that during interphase, so we just make a full copy. And then we have our sister chromatids. So after meiosis 1, so this guy, prophase, it goes in the metaphase, all of these line up in the center, and then the homologous pairs get separated, and because it's a whole homologous pair getting separated, a whole chromosome, we end up with half the number. And then after meiosis 2, we are separating the sister chromatids from one another. Okay, so here's meiosis 1, what it looks like, and then after meiosis 2, that's what it looks like. Okay, so hopefully that helped demystify the chromosome count. So meiosis and mitosis, again, so both cells are going to divide the nucleus. We're dividing the chromosomes. Mitosis is just a single division. Meiosis has two, meiosis 1 and meiosis 2. Homologous chromosomes pair to form tetrads in meiosis, but not in mitosis. Remember, we have that crossing over. So crossing over occurs. We have random assortment of chromosomes. Well, this should say metaphase 1. We have a random assortment, random alignment of chromosomes, and they're going to get sorted into random cells. So that's how our variation comes about. And then meiosis is going to reduce our ploidy count. So we're going to go from diploid to haploid. Mitosis does not. Mitosis has diploid to diploid. So remember, we start with 46, we're going to end with 23. Okay, so here's a nice little graphic that goes over the differences of meiosis and mitosis, which is brought to you by Amoeba Sisters. We're going to show you the chromosome number, and there we go. All right. So here's a nice graphic from your textbook that compares meiosis to mitosis again. Let's see. Okay, so I told you before, meiosis 1, very different. Meiosis 2 looks identical to mitosis. Okay, so you already know mitosis, so just apply that to meiosis 2. So meiosis 1, very different. The cell, or the chromosomes, line up in the middle of the cell in a double file line, which is not the same. So mitosis is just single file. Same with meiosis 2, single file line. And then the whole chromosome gets pushed to one side versus half of that chromosome. Okay, so do you see that difference there? And then at the end, there are 4 chromosomes, just like the beginning, but at the end of this one, there are only 2 chromosomes, and that's half of what it is in the beginning. Plus, there's some crossing over that occurred as well. Alright, so there's a nice video that reviews meiosis, brought to you by Bozeman Science. Alright, so let's review the stages of meiosis. So meiosis usually produces 4 daughter cells, right? 4 daughter cells. At which stage of meiosis are homologous chromosomes separated from each other? That's going to be metaphase 1, homologous chromosomes. At which stage of meiosis are sister chromatids separated? That's at meiosis 2. Or sorry, metaphase 1 and anaphase 1. Metaphase 2 and anaphase 2. In what 3 ways is genetic variation in daughter cells increased? So we have crossing over, and then we have random alignment during metaphase, and random assortment during anaphase. And then in what stage does crossing over happen? It only happens in prophase 1. Okay, so that's 11.1. We're going to go on to 11.2, so we're almost finished. We're going to explain that meiosis and sexual reproduction are highly evolved traits. We'll identify variation among offspring as potential evolutionary advantage of sexual reproduction. We'll describe the 3 different life cycle types among sexually reproducing organisms. So the way that we produce gametes is not always what you find in nature. All right, so diversity. So meiosis produces genetically different organisms, or at least genetically different egg, sperm, spore cells. Crossing over is one way we can get diversity. So during prophase 1, those homologous pairs are going to exchange segments. We have random alignment at metaphase 1 and metaphase 2. So we could have alignment like this. We can align like that. So this guy flip-flopped. Or we can flip-flop this to have it like that, and flip-flop this guy. So random alignment, and then random alignment again in metaphase 1, or metaphase 2, sorry. We can line it up like this, or we could flip-flop it and line it up like that. And then after metaphase, we'll get random assortment into our final 4 daughter cells. Random fertilization, probably didn't think of this one. So during conception, there's usually 1 egg cell that's present, but there are a lot of sperm cells that are present. So it's totally random on which sperm cell gets to the egg first. And then mate selection can be a way to increase diversity. And so random mating could bring about very genetically different offspring. Okay, so poll question. What is a likely evolutionary advantage of sexual reproduction over asexual? So let's think about asexual reproduction. So we don't get that many offspring with asexual, and they're identical to the parent cell. So the evolutionary advantage for this one, sexual reproduction results in variation in the offspring. Asexual reproduction, like, yes, it involves fewer steps. There's a lower chance of using up resources in the environment. And sexual reproduction, or asexual reproduction, would be like more cost effective because you don't have to go through an entire second process. But sexual reproduction results in variation. So it's cheaper to do asexual, but it's more, it's better, evolutionary speaking, to do sexual reproduction. Okay, so the life cycles of sexually reproducing organisms could be one of 3 things. We are diploid dominant, meaning that our main form, like your body, is diploid most of the time, correct? So the other 2 are haploid dominant, and then alternation of generation. So they switch between diploid and haploid. Okay, so let's look at these. Diploid dominant, so it's like you're the diploid dominant, your gametes are the haploid stage. So very short-lived haploid stage. All of the cells in your body are diploid cells. That's all you need to know. Okay, so here's a nice image from your textbook. Diploid cells, they make haploid cells combine to make a diploid cell. Alright, so haploid dominant, so most fungi and algae are haploid most of the time during their life cycle, and then they become diploid when they reproduce, which is opposite of us, correct? So they're diploid only when they start to reproduce sexually, but they're haploid for the rest of their life cycle. And then alternation of generations occurs in plants, and they switch between the, well, in lower plants, they switch between haploid and diploid stage. So in mosses and ferns and the like, they would have a diploid stage, and then they would also have a haploid stage, which gives rise to the diploid stage. So they just kind of alternate between the two, and they split their time pretty much evenly between the two. All right, poll question, last one. Hydras and jellyfish. Let me tell you a little bit about hydras and jellyfish. Hydras typically reproduce asexually. Jellyfish typically reproduce sexually. Okay. So an asexual being and a sexual being both live in a freshwater lake, and it's slowly being acidified by a chemical plant. Which of the two, the asexual or the sexual, are predicted to be better to cope with the changing environment? So if you said the sexually reproducing organism like the jellyfish, you are correct. So they will be they'll be more adept to coping with that changing environment. The hydra, since they're just making clones of themselves, if that clone that they keep making over and over doesn't have the good genes to cope with the acidity, then a big mass of them are going to die off, and then the population will decline rapidly. So I hope that answers that for you, and that is the end of the PowerPoint for chapter 11. It was a short chapter. So hopefully now you can explain why sexual reproduction is better than asexual. Hopefully you can identify the sources of variation in sexual reproduction, and hopefully you can describe the 3 life cycles of sexually reproducing organisms. And that is all I have for you today. Thank you.